Proteins. Proteins are the most abundant organic compounds to be found in cells and comprise over 50% of their total dry weight. They are present in all types of cells and in all parts of the cell. Proteins perform many functions. They build many structures of the cell. All enzymes are proteins, and in this way they control the whole metabolism of the cell. As hormones, proteins regulate metabolic processes. Some proteins work as carriers and transport specific substances such as oxygen, lipid, metal ions, etc. Example is hemoglobin. Some proteins called antibodies defend the body against pathogens. Blood clotting proteins prevent the loss of blood from the body after an injury. Movement of organs and organisms and movement of chromosomes during anaphase of the cell division are caused by proteins. Proteins are the polymers of amino acids, the compounds containing carbon, nitrogen, oxygen, and hydrogen. The number of amino acids varies from a few to 3,000 or even more in different proteins. Amino acids. About 170 types of amino acids have been found to occur in cells and tissues. Most of the proteins are, however, made of 20 types of amino acids. All the amino acids have an amino group and a carboxylic group attached to the same carbon atom known as alpha carbon. Or maybe hydrogen atom as in glycine, or methyl as in alanine, or any other functional group. So amino acids mainly differ due to the type or nature of the R group. Amino acids are linked together to form polypeptides proteins. The amino group of an amino acid may react with the carboxylic group of another, releasing a molecule of water. For example, glycine and alanine may combine together. The linkage between hydroxyl of carboxylic group of one amino acid and hydrogen of amino group of another amino acid release water and a carbon-nitrogen link to form a bond called peptide bond. The resultant compound glycylalanine have two amino acid subunits and is a dipeptide. A dipeptide has an amino group at one end and carboxyl group at other end of the molecule. So both reactive parts are again available for further peptide bonds to produce tripeptides tetrapeptides, and pentapeptides, leading to polypeptide chain. Structure of proteins. Each protein has specific properties which are determined by the number and the specific sequence of amino acids in a molecule, and upon the shape which the molecule assumes as the chain folds into its final compact form. There are 4 levels of protein organization. Primary structure. The primary structure comprises the number and sequence of amino acids in a protein molecule. F. Sanger was the first scientist who determined the sequence of amino acids in a protein molecule. After 10 years of careful work, he concluded that insulin is composed of 51 amino acids in 2 chains. One of the chains had 21 amino acids and the other had 30 amino acids, and they were held together by disulfide bridges. Hemoglobin is composed of 4 chains, 2 alpha and 2 beta chains. Each alpha chain has 141 amino acids, while each beta chain has 146 amino acids. The size of a protein molecule is determined by the type of amino acids and the number of amino acids comprising the particular protein molecule. Now we know that there are over 10,000 proteins in the human body, which are composed of unique and specific arrangements of 20 types of amino acids. The sequence is determined by the order of nucleotides in the DNA. The arrangement of amino acids in a protein molecule is highly specific for its proper functioning. If any amino acid is not in its normal place, the protein fails to carry out its normal functions. The best example is sickle cell hemoglobin of human beings. In this case, only one amino acid in each beta chain out of 574 amino acids do not occupy the normal place in the protein. In fact, this particular amino acid is replaced by some other amino acid, and the hemoglobin fails to carry any or sufficient oxygen, hence leading to death of the patient. Secondary structure. The polypeptide chains in a protein molecule usually do not lie flat. They usually coil into a helix or into some other regular configuration. One of the common secondary structures is alpha helix. It involves the spiral formation of the basic polypeptide chain. The alpha helix is very uniform geometric structure with 3.6 amino acids in each turn of helix. The helical structure is capped by the formation of hydrogen bonds among amino acid molecules in successive turns of the spiral. Beta-plated sheet is formed by folding back of polypeptide. Tertiary structure. Usually, a polypeptide chain bends and folds upon itself, forming a globular shape. This is the protein's tertiary conformation. It is maintained by 3 types of bonds, namely ionic, hydrogen, and disulfide. For example, in aqueous environment, the most stable tertiary conformation is that in which hydrophobic amino acids are buried inside, while the hydrophilic amino acids are on the surface of a molecule. Quaternary structure. In many highly complex proteins, polypeptide tertiary chains are aggregated and held together by hydrophobic interactions, hydrogen, and ionic bonds. This specific arrangement is quaternary structure. Hemoglobin, the oxygen-carrying protein of red blood cells, exhibits such a structure. Classification of proteins. Because of the complexity of structure and diversity in their function, it's very difficult to classify proteins into a single well-defined fashion. However, according to their structure, proteins are classified as follows. Fibrous proteins. They consist of molecules having one or more polypeptide chains in the form of fibrils. Secondary structure is most common in them. They are insoluble in aqueous media. They are non-crystalline and are elastic in nature. They perform structural roles in cells and organisms. Examples are silk fiber from silkworm and spiderweb, myosin in muscle cells, fibrin of blood clot, and keratin of nails and hair. Globular proteins. These are spherical or ellipsoidal due to multiple foldings of polypeptide chains. Tertiary structure is most common in them. They are soluble in aqueous media such as salt solution, solution of acids or bases, or aqueous alcohol. They can be crystallized. They disorganize with the changes in physical and physiological environment. Examples are enzymes, antibodies, hormones, and hemoglobin.